Okay, so this is the this video is going to be about the required practical AQA want you to do, which is looking at the effect of pH on enzyme activity. More generally, just looking at experiments to measure how fast an enzyme is working. Okay, so the enzyme we're going to look at is amylase, and what amylase does here is it digests starch. Okay, now starch is a polysaccharide. Each of those green units there is each of these is a glucose, and starch is made of loads of a couple of thousand glucoses joined together, and the enzyme breaks the starch up not into individual glucose units actually, but into this maltose here, which is actually a disaccharide. So that's two glucoses stuck together. Now, how are we going to measure the rate of enzyme activity? That's quite difficult. Now, we could look at how quickly the maltose is being formed. That's tricky. But instead, what we're going to do is look how quickly it takes, time taken, to split up or hydrolyze all of the starch. And that's there's quite a good way of doing that, is because if you remember that starch in the presence of iodine, it goes a blue-black color, whereas if there's no starch there at all, if there's just the maltose after the reaction is finished, the iodine is just going to stay its usual sort of browny color. Right, so what we're going to do is we're going to investigate the effect of pH. So our independent variable here is going to be pH. We're going to try like pH 5, 6, 7, 8, something like that. And what's our dependent variable? Well, that's going to be the time taken for all the starch to be split up. Now let's think about control variables here. Right, now we have to think about the following things. Now obviously enzymes are very sensitive to changes in temperature, so we want to make sure that they're all at the same temperature. So we're going to keep the temperature constant, and we're going to do that by using a water bath. And enzymes work pretty well at about 35 degrees C. We don't want to go too high above that. They might start getting denatured. We don't want it too low because the rate will be too slow and the experiment will take too long. So we're going to do that by putting them in every tube we use is going to go in a water bath at 35 degrees C. What else we need to keep the same? Well, we want the concentration of starch to be the same in every experiment, and the concentration of the enzyme to be the same in every experiment. And the only thing we want to change is the pH of the solution. So we've got to think about that in our experimental design. Right, so what we're going to do is we're going to have in each tube, let's get rid of that, we need to have some, we're going to have a mixture of starch, buffer solution. Now a buffer solution is a solution with a defined pH, and it won't change during the course of a chemical reaction. And also we're going to need some amylase obviously in there as well. We're going to leave that, and then while it's reacting, every 10 seconds we're going to dip in a glass rod and take the glass rod out and then put it in some iodine to see if the iodine goes black. If the iodine does go black, it means we've still got starch there. If it stays orange color, all the starch is gone. So let's see, bearing in mind how we're going to do that experiment, bear in mind we've got to keep all of those, make it a fair test. So I'm going to get set we're going to test 5 different pHs there. You can see them on the left. So we've got buffers ranging at pH 4, 5, 6, 7, and 8. So you've got 2 centimeters cubed of buffer solutions in those tubes. They all go in the water bath at 35 degrees C. And then we get 5 test tubes of starch, and we put 2 centimeters cubed of starch in every single one of those. They're all absolutely identical to each other. And another 5 tubes where we've got 2 centimeters cubed of amylase. They're all identical to each other. So we leave them all in the water bath 5 or 10 minutes, and until we're sure that all of those chemicals, all of those solutions are at a temperature of 35 degrees C. So probably a good idea to put a thermometer in one of the solutions and one of the tubes, doesn't matter which one really. And so we're sure that they're all 35. Now, when we want to start the reaction, then we take 3 tubes. So we take a black one with the buffer, we take a red one with the starch, and a blue one with the amylase, and we mix them all up. So so you're going to have a total volume of 6 centimeters cubed in that tube. You've got your starch, you've got your amylase, and you've got the buffer solution. Now that's really important, that we pre-incubate all of these 3 solutions to make sure they're all at 37. So when we do mix them, we know the temperature, we know we're not adding a cold solution where the temperature is going to go down or anything like that. Also, because we've added the same volume of, for example, starch every time, and the same final volume is 6 centimeters cubed, well, every tube is going to have the same final concentration of starch. Also, every tube is going to have the same final concentration of amylase, and the only thing that's changing is the buffer. Right, so we start the clock, get our stirring rod, put it in there. Every 10 seconds, then we take the stirring rod out, and then we just dip it in one of those wells. They're all containing iodine there, I2, which is that brown solution. Now, if there's still starch there, it will go black. Okay, and we keep doing that every 10 seconds. And obviously, we've got to give the stirring rod a quick wipe in between each, quick rinse in between every 10 seconds. And you keep on doing that until, and you'll go, it'll go black every time, black every time. That's 40 seconds, 50 seconds. And then we do it, this one, 60 seconds, it doesn't go black. It stays brown. So that means all the starch is gone. So the time taken for the reaction to occur, in that case, would be 60 seconds. Okay, now this experiment is actually very difficult to set up so that it fits into a lesson, because you can imagine if there is too much amylase, well, it's just going to digest the starch really quickly, and so you won't be able to have time to, it will digest it in 10 seconds no matter what the pH is. If on the other hand you put not enough amylase in there, you could be waiting for each one for like half an hour for it to clear all the starch, which you wouldn't have time to do all 5 tubes. So it's very tricky to set up, and quite often it doesn't work that well. Okay, so here is a set of results. Okay, so here, right, the pH of the solution in this column and the time taken for the starch to disappear. Okay, so we can see there right, so just got my calculator. Right, so the time taken for the starch to disappear with pH 4, it took longer than 300 seconds. So the enzyme is just working really, really slowly there. So we'll just leave that one. pH 5, 150 seconds. pH 6, that was the quickest one. Okay, so that would appear to be the optimum pH 6, the optimum pH for amylase would appear to be 6. At pH 7, it took a little bit longer, 170 seconds. pH 8, 280 seconds. Now, if you want to, you can actually work out the rate of the enzyme reaction, because what we've looked at there is the time taken for the reaction to complete, which is not the same thing. So you can see here that rate would be proportional to 1 over the time taken. So right, if the time is very big number, then the rate is going to be a very small number, slow rate. If the time is a tiny number, the rate is very big, it's a fast rate of reaction. So what most people do here is you can do like 1 over the time taken as a measure of the rate. So if you did 1 over 300, that would give you, sorry, we'll say this one here, 1 over 150. The trouble is that would give you a bit of a, that would give you a decimal. So it's what most people do is instead of having 1 over time, they put 1000 over time. And that gives you sort of easier numbers to work with. So for example, this one here, 1000 divided by 150 gives you 6.7. That's the rate of the enzyme reaction there. Let's do the quickest one at 6. So that only took 60 seconds. So 1000 divided by 60. That is 16.7. All right, so we can see there that rate. And you can do the same for all of the other ones there. Okay, so that is how you do measure the rate of reaction with of an enzyme catalyzed reaction, in this case amylase digesting starch, and it is quite difficult to ensure that you control all the variables. So we only want to change the pH and see how that affects the rate of reaction. So we have to keep the concentration of the starch, the amylase the same, we have to keep the temperature the same to make it a fair test.